Good afternoon. Thank you for joining us today. My name is Eugene Krunsel. I'm Chief Scientific Officer of Accelerate Health. And today we have a very interesting webinar with their hope of demystifying the FDA. And we'll talk about some very interesting new initiatives that one of the units at FDA is doing to help develop more innovations and partner with researchers and academic institutions in bringing them to be a part of the process. So a few words before we start, few words about us. As I mentioned, I'm Chief Scientific Officer of Accelerate Health, and Accelerate Health is a early stage healthcare focused startup accelerator. We're headquartered in Louisville, Kentucky, but we have a national footprint. We have a satellite operation in Michigan, and we also lead an effort funded by NIH through NIGMS STTR grant to develop and operate one of 4 regional idea states technology transfer acceleration hubs. Ours is in the southeast of the United States and covers 6 states and Puerto Rico. And Accelerate Health is also an award-winning accelerator, multiple SBA awards for support of acceleration of startup businesses, and in particular, helping with SBIR and STTR funding. So a few words about our presenters today. First, our prime presenter is Dr. Admir Garrison, who is the Director of Office of Science and Engineering Laboratories at their CDRH, Center for Devices and Radiological Health at FDA. His office is responsible for providing technical expertise and analysis and support of regulatory processes within CDRH. And prior to joining FDA, he was president CEO of a biotech startup, Ortho Regenerative Technologies, which focused on developing novel biomaterial approaches to surgical soft tissue repair. He also held senior positions at multiple companies in medical devices space. And he has an undergraduate degree from University of Oxford in biochemistry and PhD from St. George's Hospital Medical School in London. His PhD is in molecular biology. He will also be joined in the presentations today by several of his colleagues. One is Dr. Dmitriev. Anton Dmitriev is Deputy Director of that Office of Science and Engineering Laboratories. He's also Director of the Division of Applied Mechanics. And prior to joining FDA in 2012, Anton spent 9 years as a Scientific Director of the Spine Research Institute at their Department of Orthopedic Surgery and Rehabilitation at Walter Reed. And he is also an adjunct faculty member at Johns Hopkins. He has a PhD in neuroscience from the Uniformed Services University of the Health Sciences, master's in biomedical engineering from Johns Hopkins, and bachelor's in biology and chemistry from Towson University. Guys, if you are not muted, would you please put yourself on mute so I don't need to police everybody? So we hear some additional sounds that we shouldn't be hearing. Thank you. Dr. Zain Arp is also joining us today for part of the presentation. He's Director of Division of Biomedical Physics in the Office of Science and Engineering Laboratories. And his division's efforts center on identifying and investigating biophysical interactions between medical devices and the human body. Prior to joining FDA, he worked at GlaxoSmithKline and was US lead for imaging technologies, worked at Los Alamos National Labs, NASA, and as I mentioned, GSK with a focus on developing sensor solutions for various applications in space sciences, explosives, pharmaceuticals, and healthcare. Very broad experiences, and PhD in physical chemistry from Texas A&M, and 17 years of industrial experience. We're also joined by Dr. Ksenia Blinova, who's an assistant director for neurological and cardiac devices electrophysiology and modeling at the Division of Biomedical Physics. She leads the Induced Pluripotent Stem Cell and Electrophysiology Lab. She received multiple awards at CDRH for scientific excellence in the development of electrophysiology methodology. She completed her postdoc fellowship in cardiac energetics and metabolism at NHLBI at NIH, joined FDA as FDA Commissioner's Fellow in 2008, and she has PhD in physics and mathematics from Moscow State University in Moscow, Russia. And last but not least, Doran Carranza, who was instrumental in arranging that event for all of us today. Doran is Associate Director for Partnerships and Innovation in the Office of Science and Engineering Laboratories, and he leads initiatives to increase the capacity and advance the office's mission. Prior to FDA, he led the deployment of national, federal, and innovative initiatives, including NSF I-Core and IUCRC programs. For 10 years, he led business development, venture development, seed investments, grants, and startup incubation and acceleration programs in the San Francisco Bay Area. And he focuses on bringing scientific discoveries from research labs into the marketplace, and holds a PhD in bioorganic chemistry from Baylor University, and also has an MBA in strategic leadership. Now, without further ado, Ed, you're it.
Thank you, Eugene, and thanks to everyone for coming this afternoon. It's a great honor to be able to talk with you, and I'm very pleased that joined by the colleagues that I am. Big thank you to Eugene for the invite, and also for the invitation to demystify the FDA. I don't think I've had quite such a challenge before me for the next few minutes, but we'll see what we can do. Let's take the next slide if we can. The office that I'm very fortunate in leading is within the FDA and within the Center, as Eugene said, Center for Devices and Radiological Health. Just to put a little bit of perspective on that, we'll go through a little bit of what we're going to do on that. And FDA, as you know, is fundamentally responsible for protecting public health. That very often sounds quite a negative thing. The traditional view of FDA is that we're a bunch of lawyers sitting in a row that say, no, you can't. Under the leadership of our center director over the last 10 years or so, we've totally revolutionized that. We're really there for, yes, protecting the public health, but first and foremost, we're there to promote public health. And part of that is very much trying to get new devices and in vitro diagnostics out to US patients where they can do the most good. Now, the thing that we fundamentally regulate within CDRH is based around what we call benefit risk. We all know there's no such thing as 100% assurance of safety. What we have to do is to try and balance the potential benefit that any device or diagnostic will actually give to patients compared with any potential downsides. So fundamentally, that is a very, very scientific process. And that's really why we want to get much, much closer to the innovators in the entire country, because we fundamentally want to try and play the probability game. The longer we can keep people going as startups, the better it's going to be for actually feeding that product development pipeline. Unlike many other parts of healthcare, devices really is driven by small startups. Most of the innovation is coming from them and not from the large companies. So part of our mission is absolutely to try and help small startups where we can. And that really comes down to how can we help innovators? Next slide, please, Dawn. That really is the role of the office that I lead. We've got about 150 scientists. We're organized into about 20 different program areas. And those areas range anything from nanotechnology, some of the ones you'll hear about today in neuroscience and some other areas. Many of them are based around specific product types, such as orthopedics or cardiovascular products, and others are based much more around technology where that has a much more broad spread. We make a lot of presentations and things a year. The great thing about what we have at FDA is we've got fantastic laboratory facilities. We have incredibly talented scientists and engineers who work for us. And what we're trying to do is really harness that to help the innovative process as much as we possibly can. That really is the reason for why I'm so proud to be talking with you this afternoon. Trying to promote healthcare is something that we're incredibly passionate about, and particularly this area of what's broadly called translational medicine. It's something I've been very interested in for a number of years, having worked for small startups as well as big global companies. And the more that we get close to NIH, we're beginning to realize that one of the big risky parts in a lot of technology development is the handover between, say, an R01 grant to early stage venture capital or private equity funding, but also the fact that a lot of people's time in startups is dedicated to developing tools and test methods and things like that, which are absolutely vital for evaluating a technology or a product in its early stage, but actually is done multiple times. And that really is the basis of what we want to try and describe to you this afternoon, about how we're trying to partner with innovators to produce an entire raft of methodologies and tool sets that can be used by multiple companies over multiple generations of companies to really drive technology development so that it can advance into more product development. Next slide, please. Our mission is to accelerate patient access to devices and to diagnostics. And we do that through what we call regulatory science, which is pretty much what I just described to you. It has two fundamental functions. The first is to try and provide a toolkit for really helping people evaluate their technology. And secondarily, it's there to make sure that CDRH reviewers, when they're looking at pre-market reviews, never have to say, I don't know. We actually need to get ahead of the curve, so when a new technology comes in, we know how to evaluate that. We know how to evaluate the benefit risk, as we were talking about a minute ago. But we do that without asking unnecessary questions. And that is really the role of the office that I lead. And that means that we can start pushing a lot more innovation through to US patients. The red science tools that we're dealing with are what we think of as pre-competitive methodologies. That they are used for actually assessing the effectiveness of a technology or the effectiveness and safety of a new medical product, medical device. So they might be laboratory methods, they might be phantoms that are used extensively, as I'm sure you all know, in medical imaging and diagnostics. We've also been really pushing the curve out in a big way on computer modeling and simulation, and allied to that some data sets. We were actually the first organization to publish the world's first fully in silico clinical trial, where we were comparing a two-dimensional digital mammography algorithm with a three-dimensional digital breast tomosynthesis. And we actually showed through a pure in vitro in silico clinical trial that we would reach the same regulatory decision based on either a real clinical trial, which took about 6 years and had, I think, 200 patients or so. And those women were obviously getting double exposed to x-rays. We actually did the same clinical trial on a virtual set of patients at 10,000 patients, and it took a weekend instead. A lot of that sort of information is now publicly available, and we're going to go through where you can access a lot of these things. So what's in it for us? As I said, we really want to accelerate innovation and technology. We very firmly believe that if you can establish a robust technology platform, that then can spin out into multiple generations of products based on that technology. And that again is a fundamental thing about devices which are very different from biologics or drugs that we in our center don't touch at all. Devices tend to evolve rather than get a massive revolution. There are instances where you do get big step changes, but typically a lot of the devices that we actually see on the market are really evolutions of prior devices, and that is almost built into our regulatory structure as well. So that means that if we can establish a playing field of a set of tools that can be used to assess the effectiveness of those technologies, then those can be used time after time, and then you get a fair comparison between a new product and a prior one that tells both innovators and product developers and ultimately the regulators and reviewers whether that technology and product is actually suitable for purpose. I think one of the things that I'm most interested in as well is where does investment in technology and product development go? We've had multiple discussions now with the institutes at the NIH, and I think we're beginning to very much realize there is a very fertile ground for us to look at in terms of pushing forward some of these tools. When the NIH invests in SBI or STTR companies, they want them to really concentrate on making the best product they can. So we really want to try and help that and allow companies to assess how good their product or their technology is, not just how well it's tested. That leads to a lot of early de-risking for those products and technologies. And that's actually very, very good. Having been CEO of a startup, it's great to be able to say you have a technology or a product that's been assessed using an FDA-blessed technique. It really does de-risk that technology, and it attracts really the confidence in potential investors, whether they're private equity or wherever the financing is coming from. And of course, assessing products through a technique that is accepted by FDA is going to help the regulatory process at the end of the day. Next slide, please. At FDA, we recognize 3 types of regulatory tool. At the, if you like, top end of the curve, we have international consensus standards, as through ISO, ASTM, and some of the other organizations. We have a set of those that we recognize, which really tells the outside world which standards would apply to a product development. One step down from those is what we call our medical device development tools. And this is the area where there are only about a dozen or so that we have fully qualified at this point. But it specifies a method or a tool that within what we call a context of use, which is very much what you would put in the materials and methods of a peer-reviewed publication, it tells both the user of that tool and the reviewers and regulators at FDA that where a methodology is used within its context of use, we're not going to start asking questions about how it was tested. So a great example of that is a phantom that we developed for use in high-intensity therapeutic ultrasound. Within that context of use, that the method is actually used to assess the thermal heat generation from a high-intensity ultrasound. So if you use the tissue phantom that we developed within its context, then our regulators are not going to ask, well, how did you measure it? How did you this, that, the other? They're not going to come around for repeated cycles of questioning. It's accepted. It gets a rubber stamp as long as the results are acceptable. We've recently started putting out what we call our regulatory science tools. Currently, these are really a collection of a lot of the work that we've been doing for many years in this area. We've collated those now, and we're trying to push those out to get more use in the community, because we think and we know already that they're going to be enormously beneficial to people. We hope that one day the ones that are more commonly used, and we don't know which those are yet, but they will eventually mature potentially into what we call MDDTs. They will get the stamp of regulatory grade, and as they get more and more used, then they're going to allow things to flow through the regulatory pathway significantly more easily. Next slide, please. What we've done as well, as I said, we've made these now publicly available. On the last slide of the presentation, we'll actually give you the URL to the website, which has those. We're continually updating these. About every 2 or 3 weeks, we have another few coming through. Got about 110 that have already been published. And the tools are very, very, as I said, they're publicly available now. They're very diverse in what we're actually trying to attract, what we're trying to help people with. The breadth of the technology and products that CDRH deals with is massive. There's 190,000 types of product. So we don't know which of these are going to be most useful at this point in time. Next slide. We've split them into 3 areas. This is and there's no test on this, you don't have to read the busy slide. This is what the website looks like. This is a list of some of the phantoms that we've published. Next one. Here's some of the methodologies that we've done again, and let's go on to the next one as well, Dawn, if we can. And again, another set of these. I would encourage you all to look at these tools, which will give you the website at the end of this presentation. One of the things that we don't know, as I said, is which ones of these are going to be more useful. And what we do know is that the community of innovators are going to be both users of these tools, they're also going to be generators of them as well. And one of the things that we want to really encourage by giving you a couple of the examples that my colleagues will take you through is we want to encourage people to be collaborating with us to develop new tools. Those are going to be enormously impactful across the whole medtech community to, as I said, really spur innovation and accelerate that innovation going through the early stages of technology and product development. And at other times, a lot of innovators are going to be users of those tools. So building up this whole network of really pre-competitive phantoms, methodologies, and things like that, we strongly believe is going to help the entire community to really start getting some technology moving in a very fast way. It allows products then to be really compared based on how good they really are and not how well they're tested or anything else. Next slide, please. So I'm going to hand over to my colleagues right now. We have Dr. Zain up, I think, coming up straight after me. He's going to talk through some advances that we've made to give you some examples in the neurology side. Zain, over to you.
Thanks, Ed. So welcome, and I'm going to give a little bit of an overview of some of the neurological device tools that we're developing. So we do actually have a lot of work ongoing in neurological devices in areas such as biomarkers, iPSC cells for in vitro testing, and just general testing for material failure or implant failure. Today, we're going to focus on the implants in general. And this slide just goes through a variety of different failures that you might see with an implant and a neurological device, in particular the electrode failing, from broken tips to mechanical failures to the biology around that tip attacking it, causing issues. All of these are critical concerns in a neurological implant. And the fact that, you know, if I get into human with this and I have this problem, often the issue that you have is going to be found a year later or some time frame later. It's going to be irreparable. So you can't go in and do this twice. You can't just remove the electrode. And that can be a massively critical failure for the patient and cause problems with the patient's health. A significant risk whenever we go look at the aspect of reasonable assurance of safety and effectiveness. And one thing I failed to mention is this is not meant to be an in-depth talk about the different activities we're doing, but just a general broad strokes. So the next slide actually talks a little bit about some of the tools that we've actually developed and are are developing to address some of these concerns with neurological implants. So the first one on the left is an in vivo reactive accelerating aging method. And what we've done in this case is we've looked at the implants from the perspective of, can I reduce the time frame in which I need to do an in vivo study to understand that a device is going to fail due to aging and biological response. We've developed a methodology that takes this failure analysis from 6 months in a mouse brain down to 7 days by coming up with a in vivo or an in vitro diagnostic test that can be used, where you dip the electrodes down in it, it mimics the biological environment, attacks the electrodes, and as you can see in the images, you can see similar damage to these electrodes, but on very different time frames. In the case of the in vitro reactive accelerating aging mechanism, you'll see the core is wiped out, that we've had the similar effects to that as to what you would get over 6 months in the mouse brain. Significant improvement in time, significant reduction in animal usage. And this is actually one of the tools that Ed was talking about. The link to the publication is at the bottom. On the right is actually looking at this from a slightly different perspective, and that's the biological response long-term to an electrode in the brain. And in this case, we're not necessarily developing the tool per se, or the tools are not listed, i.e. we're using things like 2-photon microscopy and optical coherence tomography to image the biological response. And we're comparing that against, at the bottom would be the response of the electrodes over time. But the actual tool in this case would be more on the lines of seeing what is that biological response that I might want to see in an animal model longer term, being able to define over a time period when I need to be, when the critical points are for this biological response to the electrode, which could then dictate in a study, when do I need to take down my animals as far as time points? How long do I need to run that study to see things like dendrite reorientation or the microglial reaction? By doing it with more advanced tools, such as a 2-photon microscopy system or using optical coherence and these imaging methods, we can come up with real-time means by which to see the event occur over that time period, come up with the proper time points, and then help refine those studies through knowledge generation that leads to better studies in the future. And that's all we have on the neurological space. I'm going to hand it over to Ksenia Blanova for discussion of the cardiovascular device tools.
Thank you, Zane. Next slide, please. Let me start with technology that is near and dear to me and that enables also to develop tools that were never possible before. Induced pluripotent stem cell technology allows human blood cells to be reprogrammed into stem cell state. And this induced pluripotent stem cells, or iPS cells, can be differentiated into multiple tissue types, including heart cells or liver cells. The technology was invented in 2007 and received Nobel Prize only 5 short years after. The advantage of iPS technology is that for the first time, you have access to human heart cells that do not rely on scarce clinical sources. The cells are not associated with ethical issues as embryonic stem cells are, and today multiple commercial sources are available. So in the last 3 to 4 years, our lab, in collaboration with key players from industry, academia, NIH, and world regulatory agencies, created a body of work to validate this exciting technology for the assessment of cardiac safety of drugs. FDA co-led a large multi-site validation study where multiple labs in US, Europe, and Japan used a pre-selected set of commercially available IPS lines and single consensus protocol and blinded cells set of 26 compounds with known clinical cardiac safety record. FDA analyzed the data from all 10 experimental sites, looking at the site-to-site, cell-to-cell variability, and this led to this most recent publication on the best practices recommendation for these assays, and recommendations listed in this paper are now outlined in the FDA guidance draft, open for public comments. The revision of the guidance will affect cardiac safety assessment, providing alternative to clinical trials. So the current effort in our lab is to translate this technology to the safety and efficacy assessment of cardiac devices. The first example of the tools is for device class called cardiac contractility modulation. This is an implantable device to treat chronic heart failure in class 3 and 4 patients with normal QRS duration who are not eligible for cardiac resynchronization therapy. Once implanted, the device delivers electrical pulses to the heart in the absolute refractory period. This is some initial evidence that these pulses improve heart contractility and lead to the improvement in heart failure patient symptoms. Despite the initial evidence of the CCM treatment effectiveness, the long-term effects cannot be predicted, largely because the mechanism of action of these non-excitatory pulses remain unknown, impeding new product development and regulatory decision-making. So our aim here is to develop a new regulatory science tool to investigate CCM mechanism using human iPS-derived cardiomyocytes. On this plot on the bottom, you see the contractility recordings before, during, and after CCM pulses delivery, and we are working on extending these initial results to 3D engineered heart tissues and also into more complex models of the heart that would mimic cardiac ganglia using co-culture of cardiac and neural cells, all human. So the next medical device class that I wanted to highlight is cardiac catheter ablation devices. Ablation is used for atrial fibrillation treatment in patients who are not responsive to drugs, and the goal of the ablation procedure is to disrupt abnormal electrical signal conduction, often originated at the atrium and pulmonary vein junctions, by inducing cardiac tissue death. The conventional ablation procedures currently rely on targeted energy delivery through catheter, either heat in radiofrequency ablation or cold in cryoablation. So the standard methods have limitations, including high AF recurrence rate, long procedure time, and most importantly, rare but potentially life-threatening side effects related to off-target tissue damage to esophagus or phrenic nerve. So the new ablation modality uses short and intense pulsed electric field inducing irreversible electroporation and cell death. The procedure is much shorter as compared to the thermal cardiac ablation, and it even has a possible specificity to the heart cells, sparing the surrounding tissues. So ablation devices based on this new approach are highly anticipated by the medical community, and there are many large and small companies that have these devices in the development. But as regulators, we face the lack of knowledge related to the effect of the pulsed electrical field parameters on electroporation outcome and the solid proof of this technology tissue specificity. So in the ongoing research project, we are aiming at the development of the in vitro test for IRE ablation device safety and efficacy that will be useful for the device manufacturers and FDA reviewers. So on the bottom, you can see some initial data confirming the increased irreversible electroporation area in human iPS cardiomyocytes with increased voltage. The tool that we are developing has a potential to decrease animal and clinical testing through systematic study of electrical pulse characteristics on the ablation outcome, verification of the IRE tissue specificity, and the validation of the results obtained in vitro through large animal experiments and computer modeling. Next slide, please. Computer modeling is also in the heart of this also developed regulatory tool for the assessment of cardiac mapping devices that are used to record electrical activity of the heart during cardiac procedures in EP lab. How well a mapping system quantifies cardiac state of the patient is not trivial to assess, since we cannot confirm the system output with the direct measurements of action potentials in various locations of the patient heart. So the framework illustrated in the figure on the right proposes an elegant solution. The device manufacturer provides FDA with the geometry of the mapping catheter, and computer model of the heart developed at OSAL simulates the electrogram signals for specific heart anatomy and cardiac status. And then we can compare the device output that is generated in a blinded fashion to the simulated condition and assess this device accuracy to make a regulatory decision. Finally, the last tool is developed to assess how a cardiac device like left ventricular, sorry for the typo here, assist device or implantable blood pumps may interact with the patients, particularly causing blood damage through hemolysis and thrombosis. These events are primarily dependent on shear stress, strain rate, and turbulent energy dissipation in the fluid, which can be approximated based on the velocity fields obtained from experimental flow visualization or computer fluid dynamics, or CFDs. CFD testing results are increasingly used in regulatory submissions, and we need credible experimental data for CFD validation. So for the two benchmark models shown on the bottom, the results of such experimental studies from multiple independent laboratories are now publicly available for CFD models validation and are already used by the industry while impacting FDA's guidance and standard development in this area. You can check out this database hosted by the NCI at the link below. That concludes my part of the presentation, and I'm happy to give it to Anton Dmitriev, who will be talking about orthopedic devices.
Thanks, Ksenia, and good afternoon, everyone. In my presentation, I'm just going to highlight a couple of examples from the orthopedic program portfolio that highlight the tools that we've developed over the years, not just in isolation at FDA and OSAL, but in collaboration with both the clinical and academic community across the nation. Next slide, please. I'm going to start off highlighting this tool that we developed in partnership with our clinical colleagues as well as several universities, and this has to do with the development of a new test method to evaluate spinal deformity correction devices. These devices are used in pediatric applications for a condition that is known as early onset scoliosis. It affects children. It's a genetic disease, and it affects newborns and children under 10 years of age. The condition is associated with very high morbidity and, as a matter of fact, mortality. And the challenge with these devices is that while the surgeon attempts to correct the deformity and allow a child's pulmonary function to resume and actually the lungs to function to begin with, the surgeon cannot fuse the spinal column together because obviously the child is actively growing. So the devices that are implanted have to periodically, at a frequency of about 2 to 3 times a year, be adjusted, and the child will have to go additional surgical procedures to allow for increased length of devices in accordance with spinal growth. And what happens is with these devices, the frequency of failure in clinical use is quite high, just because the overall load and the fatigue fatigue capacity of these devices sometimes is exceeded. So what we've done, we've partnered, in this particular case, we've partnered with clinicians, and we analyzed some of the failures of these devices that were used in the real world setting. And we created computational models that predicted the high stress concentrations around these devices, and then identified long-term fatigue testing parameters that would enable a more accurate prediction of their performance in the clinical setting. So through this collaboration, we're able to now draft actually a new standardized test method that is now undergoing review through the ASTM framework in order to help manufacturers create devices that are not only expanding the surgeon's portfolio of armamentarium that can be used to treat this debilitating condition, but really enhance their safety profile and enhance the performance of the devices, and therefore reduce the number of complications and postoperative failures. Next slide. And another example, it's in a rather similar setting, and as it is highlighting a new test method that we developed for spinal device performance evaluation. However, this is to treat a condition that is predominant in the adult population. This is a condition known as degenerative disc disease. It's very prevalent. It could be also quite debilitating. It's one of the top causes of loss of work and loss of productivity in the United States. And one of the more recent innovations in this space has been the development of the intrabody fusion spacers, which you see on the right-hand side, that incorporate into integrated fixation features such as screws or attached plates. And what happened was with the preclinical evaluation of these devices, it was not uncommon to not adequately analyze the fixation strength of these features, such as screws, in the vertebral bone. So our scientists developed a robust cadaveric fatigue testing protocol that is aimed to predict more or less the fixation potential of the integrated fixation features to really assess the long-term performance of these devices before the actual biologic fusions occur. This hopefully will allow the stakeholders to utilize the method and test their devices in vitro, and therefore still obviate the necessity for any clinical trials, because the in vitro cadaveric work could be sometimes in particular settings predictive of clinical performance. And I think that's all I have for today. So next slide. I will probably be turning it over back to Dr. Margerson.
Thanks, Anton. I'm going to bring it home now to make sure that we've got plenty of time for a discussion and questions. Just one other last example before we wrap it up. The virtual family, next slide, Dawn, please, is a set of computer models that we developed with the ITIS Institute in Zurich. And we've got MRI maps and anatomically correct whole body models now, along with a lot of the tissue properties, including thermal, electromagnetic, etc. These are again are publicly available. We've got multiple different people, different BMIs, different sexes, etc. And this was used, next slide please, Dawn, a number of years ago, just over 2 years ago now, where we gave clearance to what at the time was the largest magnet used in an MRI. It's a 7 Tesla magnet. It came from a company called Siemens. And this is a quote that's taken from their website. And the bit I really just want to point out is that the use of the virtual family that we developed allowed them to get clearance on the 7 Tesla magnet in their new MRI machine without needing any clinical trial or any preclinical animal models, because we could measure the specific absorption rate coming from the radio frequency and magnetic fields purely through modeling. So the impact for the company was dramatic. And this is clearly a large, fairly dramatic example of it. But it shows how that we can de-risk some of the product and technology developments, and we can really speed it up. By using these sorts of models and tools that my colleagues and I have described, all the way through product development and technology development processes, we think we can have a dramatic effect on getting more technology to market. And as I said, playing the probability game. Next slide, please, Dawn. Our ideal really would be that we could partner with all of the innovators and startups across the country, but clearly bandwidth doesn't allow us to do that. And that's one of the real reasons that we want to try and make a lot of these toolkits as available as possible. It carries with it a certain confidence that the FDA is aware of these ways of measuring and assessing technologies. And I think that really will help the community. As I said, we've got over 100 of these now published. And I think the whole community will either fit into being the users of a reg science tool or the developer of a regulatory science tool, and will potentially play different roles depending on what they're doing at any particular time. What we need to find out is what are the additional regulatory science tools that are going to be critical to accelerating the most important medical devices that we're going to see coming through in the next few years. It's very difficult for us to predict that. What we want to do is to work very, very closely with the whole community, that we can have collaborations to start developing more of these tools. We can publicize them, and that allows more and more people to focus on how good the technology is and not how well it's being tested. Next slide, please, Dawn. So we're going to finish it up there. I'd like to thank you all for logging in. Again, thank Eugene for the invitation to be able to talk to you this afternoon. There's 3 websites there that we'll leave up while we're having a discussion, or they'll be available either through us or through Eugene's organization. The first is our email address. Please do feel free to email us any particular questions that you may have. I think our email addresses are also public information on the FDA website, so please do feel free to look us up and contact us direct as well. Second one is the website for the office, which has a broad overview, and you can obviously drill into different bits. And the third URL there is the link, the direct link to the top page for the regulatory science tools. So Eugene, we'll stop there and invite any questions or comments that people have.
No.
Fantastic. Thank you so much. I think that, I already mentioned, but that webinar and all the slides will be available to all the participants, and will be available on our website. So all the contacts and all the links. If anyone has any questions, please, put them in chat, so we have in order of getting them asked. While we're waiting for any of those to come in, I'd like Ad, you maybe to expand on how exactly would you envision that collaboration, how researchers and startups that are part of our network, that are part of other proof of concept networks, and the university that we work with and startups that we'll work with, how they can promote to their researchers, how they can come and work with you. And that was the whole point, how we were hoping to get people to feel a little bit more open, working with you and demystifying FDA in that, not in terms of necessarily how to get your device, approved by FDA, but how can we work with FDA so we know, people there will get to, get introduced to the right people. How can we partner with your organization?
Yeah, it's a massively important question, Eugene. Thank you. We've traditionally had many collaborations very broadly across the whole of academia. They've been enormously profitable for us and incredibly useful. One of the things that we're doing now is we're getting a lot more open about these are the areas that we're really working in. And over the next couple of weeks, we're going to be updating our website with the specific areas that are the big problems we're trying to solve at FDA. We've talked to people like National Science Foundation and people like that as well. And one of the things that we're finding very much is that people really want to work on problems that are considered important by the FDA. I'm a relative newcomer to FDA, so I'm constantly still surprised about how people want to work with us, and they really do. So one of the things we're doing is we're setting our agenda and saying these are the big goals that we think we're trying to tackle, and we invite specific collaborations in those areas. Unlike NIH, we don't have a big checkbook at all. But I think people very much, firstly, want to work on something that's important. And the other thing about a lot of these regulatory science tools that we're working with some other organizations now who are very much going to be developing tools that effectively they're going to get intellectual property of some shape or form on this. And they're actually looking potentially to start a market in that as well. So for example, with my old hat on working for a large company, if I was doing some sort of pre-market testing, and I could do that with some sort of test methodology that I knew had the blessing of FDA, then I would do it, even if I thought it wasn't the best test for my device potentially, or I didn't like the test. I'd do it, and I would write a check to actually be able to use that. So we're really inviting two communities to work with us. One is the developers of tools, and that can be just for the sake of scientific merit in its own right. Other people will be developing tools specifically to generate intellectual property and potentially benefit from that in the future. But we're also really interested to work with people who are using the tools to actually inform us how well they work. Because just as medical device technology is evolving and accelerating all the time, so is the science of assessing those products and technologies. And that's really the part that we play. I didn't mention earlier on, I don't think, Eugene, that of all of the work that we do within CDRH, the people in our organization in OSAL take part in roughly 1/3 of the pre-market clearances and approvals, the pre-market reviews. So the closer we can get to the scientific community and make sure that the science of assessing devices and technologies keeping pace with the science of accelerating that technology, then we're all going to be in a much better place. I would invite everybody to keep a very close eye on our website, the link that you said you're going to share. As we evolve that literally over the next couple of weeks, there's going to be very specific areas that we're saying, these are the areas that we're seeking collaborations in. And part of that website will also have all the right direct contacts for us and things like that. So we openly invite that.
Fantastic. That's that's very helpful. We let me see what we got. Jackie, you had something you wanted to ask about this, the startups and our community and SMEs.
Yes, I was just starting to type it, Eugene. Sorry about that. Our researchers and innovators really need to connect with folks in the market to understand the different processes related to commercializing technology. And FDA regulations is really important to them, as is reimbursement. Can you talk about how innovators might be able to access folks in the FDA that could further their understanding of the regulatory process?
Oh, I'd be thrilled. That's a yes. I think that's a really important area. First thing to stress is that, and I've been on both sides of the fence, the old scary FDA is gone. We are very much there to try and help innovators and get technology through. That's our job. Our job is not to stop good technology getting to US patients. It's to do what we can to do that. So there are several ways that can be done. One of the things I draw people's attention to is an organization in CDRH. It's called DICE, D-I-C-E. That's the Division of Industry and Industry Communication and Education. I apologize, DICE. Again, very easy to find on the website. They are specifically there as a concierge to help people get their questions answered. So you can go to them. They will either direct a question to, if it's a technology question, it would come to someone in OCEL typically. If it's more of a regulatory product development question, it's more likely to go to one of the pre-market offices. But they will act as a concierge to get that question answered. If it's a technology question, we're quite happy to try and entertain that directly through OCEL. So those are the easy, quick ways that you can get some quick advice, quick answers, and things like that. We're actually looking to broaden the number of specific technology webinars that we're doing publicly as well. If you actually want some more, if you like, official advice on a regulatory question, the best thing to do is go through what we call our pre-submission process, which allows you to have a meeting with our reviewers, either probably remotely these days, of course, but that will allow you to put effectively written questions and get written answers back, and you will get some very specific regulatory advice. So it spans this whole spectrum from, you can get a quick question answered, hopefully through DICE or through ourselves, or go up to the pre-submission process.
And sometimes if it is, maybe if it would be possible to think about when you don't, your question is not quite at the level of, pre-submission, meeting, but maybe it's a 20-minute conversation or 15-minute conversation. Do you think that, and I don't want to put you on the spot, but maybe, you and your colleagues, that we can, think about sending people to, from, it will not be very often, but from time to the question. Would that be okay?
Yeah, absolutely. And I think, especially if it's on the real sort of technology development side, where we start getting into more regulatory questions, we might be able to help. We probably need to bring in some of the full-time pre-market reviewers, so that may take a little longer. The other thing is actually the resources on the FDA website are actually pretty impressive, to be honest. There's quite a lot there about what's the difference between class 2 and class 3 devices and things like that. A lot of those basic things are very well described on the website. I'd encourage people to do that, certainly. But we're always happy if we can, if we have the bandwidth to answer questions quickly, we're very happy to do that. Especially because it kind of gives us an idea of where people are finding the whole process quite difficult. Because if we can find out where things are difficult, that's an area that we can try and solve and make more easy for people.
Fantastic. That's exactly, that's exactly it. Yeah. Yeah, we have a question. Catherine Ha, do you want to ask that question, directly? You can unmute.
Yeah, sure. So we're trying to do user testing of our device and trying to, test and then see what works well and then fix it before we really get a FDA product that we want to get approved. And I was just wondering, is there anything that we should be aware of regulation-wise when we are conducting these usability tests? I know that we can't market it because or sell it because it's not diagnosing anything because it's not FDA approved. But beyond that, is there anything we should be aware of while we're doing these user tests, usability tests for a medical device?
Well, I think that I'm going to give the classic regulatory answer. It depends. There are so many variables in these sorts of things. To be honest, Catherine, I think that would be a great, it's difficult to answer without specifics. And in a public forum, it can be difficult to get down to some of those specifics. I think this is a classic area where a phone call to DICE or an email to DICE would be really well worth it, because if you can be a little more specific on the device, how you're looking at it with the user groups and things like that. In theory, user groups are a great way really to feed into getting towards a design freeze, which sounds like what you're trying to do.
Exactly.
So there's no, really a lot of the time, FDA is going to look at something post-design freeze, because we really want to, we assess a product in its final marketed form, fully sterilized, fully packaged, whatever it is. So we're interested in how you reach that design freeze, of course, what information goes into doing that. But there's no real sort of overarching regulatory pathway there. But I think this is a great example of go to DICE, you should be able to get the question answered really very quickly.
Mm-hmm. And on the side note of that, for like artificial intelligence, I know the FDA is adding more regulation on trying to figure out how to keep up with the continuous development of artificial intelligence algorithms. Do you know much about that process of back and forth with the FDA and then the developing technologies?
Yeah, I mean, we're going to or have recently published a white paper on adaptive algorithms and AI and things like that. For CDRH, AI is a really interesting one because it's been built into devices, particularly diagnostic devices, for many, many years. So we're very familiar with how we actually evaluate that. As you say, there has been a massive acceleration recently, and we're seeing it coming into the fields of digital pathology and all sorts of other areas as well. But at the end of the day, we don't regulate AI, we regulate the device. What's made it more complicated over the last few years, particularly with this huge acceleration, is that we now recognize that there's different categories of device. So we have what we call SAMDs now, which is software as a medical device. We also have SIMDs, which is software in a medical device. And there are nuances as to how those are regulated. Over the last 12 months or so, we have published about 3 guidances with a capital G. Guidances, for those of you who don't know, are how we make our policies publicly accessible and publicly available. So these are guidance documents that, while they are not legally enforceable, it's a very good idea to follow them. And we've updated 3 of those ones. For example, clinical decision support software is a recent one that got published. So my advice on that one would be check out the guidances first, because it's evolving very, very rapidly. But we don't regulate AI per se, we still regulate the device.
Okay, so if the device was already, like say we had a device that was regulated already, and we're putting AI on top of that, it should be okay to use theoretically?
I can't answer that without all specifics, Catherine, I'm sorry.
No
there is the other guidance I would point you to is whether that would be considered a modification of the device.
Okay.
That's a guidance that it literally is called 510K modifications or something similar to that. That defines the sort of the threshold, the borderline, which if you cross over, then would mean that a new pre-market application would be required.
Thank you very much for your answers.
My pleasure. Hope it helped.
Thanks, Ed. Stan Sansone, you had some comment? He may have left. You said thank you for recognition of small businesses' value to the health of the nation. So that is something that is really, I think, is really important, as what you mentioned, Ed, about the ways how not only large universities can partner with you guys with FDA, but also the value that small businesses bring into that whole process. So it was very generous of you to provide your contacts and offer people. I hope you will not regret that later on.
I won't regret it. There is an interesting fact, Eugene, that many people are not aware of. The majority, like that, it's very easy to sit here and say small businesses are really the heart of innovation, but it's factually correct as well, because over 90% of people in the medtech industry work in companies of 50 people or less. It's not the big global enterprises that myself and many other people have worked for that are really the heart of innovation and the heart of pushing things forward to the next generation. It really is the small startups. So I really do see there is this pathway from R01 grants from the NIH to then forming an SBIR or STTR company, to then that trying to get some private equity or venture capital funding. And depending on what the exit strategy of those entities are, that is actually the fuel that feeds the process of innovation for medical devices and diagnostics in this country. So that's why we're trying to focus on all areas from the FDA perspective. We're trying to work closely with CMS to make sure that reimbursement is somehow parallelized with the product development process as well, because they're unhinged at the moment. We all know that. But we're looking at all parts of this innovation process and seeing what we can do right from the early innovation, which is where my interest is, all the way through to actually having a commercially viable and, more importantly, clinically successful device.
Fantastic. That's exactly what Jackie was talking earlier about. Those two areas, regulatory and reimbursement. Those are two critical areas, for startups in particular to get to know more. Ken Dicken has a question.
Yes, Eugene, thank you for recognizing my hand up. Eugene, I want to thank you for relieving some of the stress. I'm a serial entrepreneur, small businesses, a lot of them. And then I'm also an angel investor. And in many of the cases, if it has to do with, we have to meet with the FDA to get something accomplished, we tend to want to raise more money. We tend to want to extend products, innovations further out. To hear that you really want to work with us in startup, small business areas is very, very welcoming to me. And I wanted to verbalize this, and I appreciate your efforts. And I certainly hope that the administration, current and future, continue with your mission. Let's just put it that way. Thank you very much, sir. Appreciate it.
Well, thank you, Kenny. And it's really important. It's early days for us with this sort of new, not new attitude, but really trying to improve things. And we're going to make mistakes. We'll get it wrong at times, but we know it's the right thing to do, because one of the big strategic targets that we have through CDRH is trying to make sure that new technology and new products are introduced to the US earlier than they typically have been. The company that I used to be CEO of a company a few years ago, and when I took it over, the strategy was, well, let's go to Europe, let's get approval in Europe, and then we'll generate money so that we can get approval in the US. You've seen that as well multiple times. First thing I did was to say, no, that's wrong, because actually the business plan doesn't work if you do it that way round, because you can never necessarily generate enough revenue in Europe because of the disjointed reimbursement in Europe to actually make it work. So the first thing I said to the board of directors at the company I ran was, well, let's change this around. Let's go to the US first. The barriers are actually not higher in the same way that they used to be many years ago. There are examples where it is. But if you can actually get into the US market, it's a better business model anyway, on the whole. And if you can do it earlier, then that's so much better. So the more we can actually really catalyze that transition, and it is a deliberate and public strategic priority for CDRH, then the better it is for US patients and ultimately for patients all around the world. Because if there's one thing I've learned, and it's a very scary thing, people do look to the FDA for leadership, which means that we've got to lead in these areas. It's just the right thing to do.
You're right. There are many smart people in the FDA, and but to a startup on a tight budget, it is scary to think we've got to approach the FDA. But let's go someplace else and get some money, get some revenue streams coming. And I don't like that, because we're actually delaying the introduction of something that could be very useful to patients around the world, saving lives, making communities better, just everything about it. And we won't need to elaborate it, Edward. I'm just very pleased to hear you say these words that you're saying, and I just hope that it's true, and I hope I got enough time to partake in that after many attempts and many dollars. Thank you again. I appreciate you. You stay safe, sir.
Fantastic, guys. Really, really fantastic exchange. Very illuminating. Thanks, Ken, for bringing it up, and thanks, Ed and Ksenia and Anton and Doran and Zane for coming to our webinar today and spending time with us and presenting, and most importantly, opening your doors and phone lines and emails for further communication. As I said there, that webinar will be placed on our website. So thank you so much. You'll hear from people who may not have spoken yet during the webinar, and some I know that because of spring break, some of the faculty members that and grad students were not available to join us today, but they asked about access to that presentation on the recording. Thank you so much. I really appreciate that. And we will, I think we'll conclude, with this.